Good afternoon. I am the expert Sergey Somov. I continue to introduce you to the inverted biological microscopes MAGUS. In the previous video, we reviewed the MAGUS Bio V300, and today I will talk about the MAGUS Bio V350 model. The fundamental differences between an inverted microscope and an upright one lie in the design of the stand. The inverted optical system and inverted lighting system allow for the examination of live specimens directly in specialized laboratory containers with nutrient media. Colonies of bacteria, tissue culture cells, or liquid sediments are found in petri dishes, terasaki plates, high flasks, or roller bottles. The design of an upright microscope does not allow for working with such specimens. An inverted microscope is versatile. It is suitable for working with laboratory containers, as well as for studying samples on microscope slides. Inverted microscopes are equipped in microbiological laboratories, scientific institutions, and IVF clinics. Such microscopes are also used by pharmacists, virologists, toxicologists, and representatives from many other fields, including agriculture. Look at how the inverted microscope is arranged. The light source is located at the top, in this part. A beam of light, limited by the field diaphragm, enters the condenser, passes through the specimen, and reaches the objective. The objective forms an enlarged image of the specimen, and mirrors direct this image to the eyepieces or to a digital camera. In general, it's all the same as with an upright microscope, just in reverse. This, in turn, is responsible for the ability to place special containers on the stage and observe objects through their bottom. The thickness of the bottom should not exceed 1.2 millimeters. Let's take a closer look at the main characteristics and design features of the MAGUS Bio V350 model. We will start with the visual attachment. The optical tube length is infinity. This means that the microscope is equipped with objectives designed for infinity, an interesting feature of the attachment. During the assembly of the microscope, the user adjusts the tubes and selects the height of the eyepieces according to their height. Type of visual attachment: Seidentopf. The tubes rotate smoothly around the hinge for easy adjustment of the interpupillary distance, which varies from 48 to 75 mm. This range takes into account the characteristics of users with both narrow and wide interpupillary distances. The standard configuration includes 10x eyepieces with a field of view of 22 mm. The diopter adjustment of one eyepiece helps to customize the diopter to the user's vision. The eye cups protect against lateral light. The visual attachment is trinocular. A digital camera or monitor is installed in the vertical tube with a visualization channel. The light flux on the trinocular attachment is switchable. There are two positions. In the position where the vertical visualization channel is closed, all the light goes to the eyepieces. In the position where the visualization channel is open, 50% of the light goes to the eyepieces, and 50% goes to the visualization channel. Thus, you can observe the specimen both through the eyepieces and on the monitor simultaneously. The second visualization channel is located next to the objectives on the microscope body. This knob switches the light flow. In one position, the light is directed to the eyepieces, while in another, it is directed to the side channel. This switching method ensures that the brightness of the light source is sufficient for use with each objective, not only for the bright field method, but also for the phase contrast method. The second interesting property of such a light distribution is the absence of parasitic illumination. Only the image of the specimen is transmitted to the camera. Light entering the eyepieces from external sources will not pass into the visualization channel, and reflections from ceiling lights will not spoil the photograph of the specimen. You can choose one of two channels or use both for different cameras. For example, in one setup, install an HDMI camera with a monitor, and in another, a USB 3.0 camera. Now let's consider the lenses. A revolving nosepiece with 5 objectives is installed under the table. The objectives in the nosepiece are plan achromats with infinity correction. The specimens under investigation live in special containers, which is why both the stand and the lighting system, as well as the optics of the microscope, are designed to work with these containers. The microscope uses objectives corrected for a bottom thickness of 1.2 mm. The revolver is equipped with 3 bright field objectives with magnifications of 10x, 20x, and 40x, and 1 phase objective with a magnification of 10x. The free slot of the revolver is designed for a 5th objective. Additional configuration with a 4x objective or phase objectives of 20x and 40x is possible. The planochromatic objectives correct spherical aberration for 1 wavelength of light, chromatic aberration for 2 wavelengths, and correct field curvature by 90%. The microscope's illumination device allows for the adjustment of lighting according to the Köhler method. Proper adjustment according to Köhler enhances the quality of the image. Under such lighting, the resolution limit is achieved for each objective. The field of view is evenly illuminated without darkening at the edges. The specimen is placed in focus, and artifact images are removed. The illuminator is securely fixed to the microscope stand. The light source is a 12-volt, 30-watt halogen lamp that is adjusted relative to the optical axis, and the collector lens using these 3 screws. The halogen lamp emits light that is comfortable for the eyes with a specific color temperature. The 30-watt lamp emits sufficient brightness for use with all objectives, both bright field and phase contrast. The illuminator is equipped with a collector featuring a field diaphragm, which is adjusted depending on the magnification of the objective lens. An important part of the illumination system is the condenser device. The condenser bracket is mounted on the stand. We move it along the optical axis using this handle. The MAGUS Bio V350 microscope operates using bright field and phase contrast methods. It is equipped with a journey phase contrast condenser that has 4 positions. A free aperture for observations in bright field, 3 apertures with annular diaphragms for use with phase objectives at magnifications of 10x, 20x, and 40x. The phase rings are centered. The diaphragm change is performed by rotating the disc until it locks into place. A number appears in the condenser housing window, corresponding to the magnification of the phase objective being used. The number 0 corresponds to the free aperture. Quick and intuitive switching from one research method to another saves time and simplifies the work of the researcher. Beneath the rotating disc is an iris diaphragm, which allows for the adjustment of a specific aperture for use with bright field objectives. To achieve greater contrast, one can use light filters included with the microscope. The light filters are installed in the hinged frame of the condenser. Center the condenser in the optical axis using these 2 screws. When working with tall containers up to 165 mm, the condenser device is moved aside. This handle is used to secure the device. Focusing. The coaxial knobs for coarse and fine focusing are positioned on both sides of the stand for right and left-handed use. The focusing mechanism moves the revolving device with objectives vertically. Fine focusing is required for precise focusing on the specimen and for refocusing the microscope when changing objectives or containers. The coarse focus locking mechanism is used to expedite the readjustment of the microscope when changing the specimen being examined. The mechanism for adjusting the stiffness of the coarse focus will help set a comfortable stiffness for each user. Another parameter of the focusing mechanism is the division value of the fine focus. On our microscope, it is 2 microns. For a laboratory microscope, such a step is optimal as it ensures both speed and accuracy of focusing. The stage. A special mechanism is installed on the stationary stage for moving laboratory containers in 2 mutually perpendicular directions. The mechanism moves the Petri dish smoothly, ensuring that the user does not miss any part of the specimen being studied. The movement handle is long, allowing the user to avoid straining the wrist of their right hand. The microscope kit includes 3 holders for Petri dishes of different sizes. The microscope is heavy, so a convenient handle is provided for carrying it on the stand. You won't have to worry about how to comfortably carry the microscope when moving it from one place to another. We offer a range of accessories for the microscope. The additional 4x objective will provide a large field of view for the quick detection of interesting areas of study. Phase objectives with 20x and 40x magnification are used for identifying non-contrasting objects measuring 0.7 and 0.4 microns. The eyepieces expand the magnification range of the microscope. An additional pair of eyepieces will help achieve useful magnification on the objective, which is more commonly used in practice. Digital cameras display the image from the microscope on a monitor and save the information as files. The software precisely measures objects in real time and edits the image. The calibration slide works in conjunction with the eyepiece, with a scale, or with a camera program. The MAGUS Bio V350 microscope is not as straightforward to use as the MAGUS Bio V300, but it is more convenient in operation. Come to our demo room, bring your samples, compare both models, and choose the one that suits you best. We will help you select additional accessories and a camera. See you soon.